Okay, so we are going to start on RNA metabolism. We're going to discuss first transcription, RNA synthesis from DNA, and we'll be discussing prokaryote, eukaryote, as well as talking about the different RNA polymerases, their function, the process of transcription. So in general, DNA and RNA are going to be similar, however, there are some very key differences. We know that at the 2' sugar, carbon on the sugar for DNA, we have hydrogen, which is very stable in an aqueous environment. And on that same 2' sugar carbon, we have a hydroxide in our RNA, which is going to be very unstable. So our ribose sugar is more unstable than our deoxyribose sugar. In DNA, adenine is always going to pair with thymine, and in RNA, adenine will pair instead with uracil. Structure of DNA is always going to be double-stranded, whereas RNA will be single-stranded. And DNA, the whole purpose of this is to store and maintain fidelity with respect to information and make sure it gets passed on from generation to generation. Whereas in RNA, its whole purpose is to translate that information stored in DNA in something functional that a cell can use. RNA also are going to have different activities, such as catalytic activities. There are different types of RNA, whereas there's only one DNA. RNA can be our traditional messenger, ribosomal, or transfer RNA, or our regulatory RNAs. So in general, RNA is going to be made up of all the RNA in the cell at any given time, and this is given the name of a transcriptome. We know that in the DNA, small regions are going to encode for actual proteins, and these are our exons. A lot of the regions that are not coding for proteins, it's been discovered, are coding for RNA, but not the traditional RNA, not the messenger, transfer, and ribosomal RNA, but rather the more regulated RNAs, or the ribozymes, the enzyme RNAs. So very briefly, let's talk about the differences in polymerases. So RNA polymerases in bacteria is only one. They have a single RNA polymerase. How they are able to control different genes is through what they have are sigma factors. Sigma factors are kind of their transcription factors. And in E. coli, they have many sigma factors. And so eukaryotes have a different approach. Eukaryotes have 3 different classes of RNA polymerases. The first class of RNA polymerase is going to be responsible for for generating all the ribosomal RNAs. RNA polymerase II is going to synthesize all messenger RNAs, so all of our proteins and enzymes encoded in our DNA will be transcribed by RNA polymerase II. RNA polymerase III is going to be responsible for our tRNAs and also our 5S RNA. Both RNA polymerase II and III can do our small nuclear RNAs and our microRNAs. So these are more of our regulatory or our enzymes. Now this is just a picture of a gene, and so some things to keep in mind with respect to this are that our polymerase, in order to transcribe a gene, needs 3 things. So our polymerase is going to need our active bases. And this is going to be A, G, C, U. It's going to need magnesium. And it's going to need a template, which of course is going to be our DNA. But it has to have access to that template. Now this is a gene, so let's just talk about some areas. We have an area that's called the transcribed region. So this is where the gene actually is. And we have an area that's called the 5' flanking region. And this is the area that can have promoter regions, enhancer regions. It can be the enhancer can be very far away from the actual gene, or it can be close to the actual gene. In the promoter region, we're going to have some consensus sequences that our transcription factors can identify so that they can bind to and activate a gene. Those similar or those consensus sequences are going to be in the form of a TATA box. It's also called a Hogness box, or a CAT box, C-A-A-T box. Now, in our transcribed regions, we are going to have positive strand because this is going to be upstream of the start of the gene. And our promoter regions, our flanking regions, are going to be negative because these are considered downstream. So downstream are going to be negative, upstream are going to be positive on the numbering of our genes. Now, in our positive sense, we have a cap site, we have a polyadenylation site. In between those, we're going to have our start, ATG, and then one of 3 stop signals. We are also going to have introns mixed in with exons. So our first messenger RNA is a heterogeneous nuclear RNA, and this is going to encode all introns and exons. Our actual messenger RNA, which will be used to make our protein, will have all of the introns spliced out and will only contain exons. Now, the only other thing to add here is that we have our promoters that are going to turn on a gene are always going to be cis-acting. So we have cis-acting promoters. And cis just means on the same strand. Trans would be opposite strand. Okay, so now let's talk just very briefly about our bacteria genes. So our bacteria genes are very simple. They have no introns. They don't have space for them. And they really don't have time to make multiple transcription factors, multiple sites for their sigmas to bind to. And so what they have are cistrons. And a cistron is simply a region of DNA that encodes a polypeptide chain. Now, for our prokaryotes, they like to link together proteins that work together. And so they're called polycistronic messenger RNA. So it contains information for several proteins. So from this polycistronic RNA, you can get protein A, B, and C. And they all are usually linked to a similar function. Now, our RNA polymerases are going to be very large molecules, and they're going to take up what we call a very large footprint across the DNA. And in fact, they are going to bind to about 35 base pairs. Now, in that is, of course, going to be that start site, which you will have an opening of your DNA to allow your polymerase to bind to. Now, the polymerase is going to bind to DNA at both ahead and behind. And you're going to introduce strain in the DNA molecule that topoisomerases are, of course, going to relieve. So we're going to have downstream negative supercoils, which means that your DNA is underwound, and then upstream positive supercoils, which means your DNA is going to be overwound. And again, this is all just to allow for the RNA polymerase to get in there and transcribe the gene of interest. Now, there are some things we need to discuss about the coding strands and the template strand. So one thing to keep in mind is your template strand is transcribed. So remember, RNA needs a template. So the template strand is transcribed. And what that means is the coding strand is identical in base pair sequence to the messenger RNA. I'm learning how to use this. And of course, U instead of T. So again, we have our template strand. Your RNA polymerase needs a template. It's going to use that DNA template strand. And this template strand is called the antisense strand, and the coding strand is called the sense strand. So your messenger RNA is sense strand. And then the sense strands are going to be identical. Now, genes are going to be written left to right. And the left is 5' to right, which is 3'. And the codons, of course, are going to be in 3 bases that will encode for some amino acid that will be strung together to form some protein. And when you're going to give a gene sequence, the sequence of gene X, you're always going to refer to the coding strand. Which is at 5', 3'. Now, with respect to replication and transcription, there are some similarities. You are going to have to need polymerases, and you have to have a template. Now, there are some differences. So let's talk about DNA. DNA is going to need RNA primers. DNA is going to move about 1,000 base pairs per second. DNA has a high degree of fidelity. They're going to have proofreading. They're going to have repair. RNA has no primers. Oops. So RNA will have no primers, about 100 base pairs per second. They have no proofreading. So if you have a mistake, it gets degraded. You can't go back and fix your messenger RNAs. Okay, so this is an example of E. coli, what's called an ORI-C site. An ORI-C site are a series of consensus sequences, and in this case, this is going to be recognized by a sigma 70. So sigma 70 is going to bind to the ORI-C site and bring the RNA polymerase to that ORI-C site so that the RNA polymerase can begin to translate this region of DNA to make the RNA molecule. So the ORI-C site, again, is always going to be preceding the start site, so they are upstream of the start site. And this is a very common thing that E. coli uses, these consensus sequences. So in E. coli transcription initiation, you're going to have your RNA polymerase. Well, you'll have your DNA, of course, with its promoter region. And you'll have your RNA polymerase, which is this very large protein. And here we have our sigma 70. And so what basically happens is your sigma 70 will bind to that promoter region, will guide the RNA polymerase to that promoter region. And this is going to be a closed complex. It's not yet available for transcription. Now after this, your sigma is going to move out of the way and allow your RNA polymerase to have access to the DNA template at this promoter site. And this is going to be an open complex. Now after about 20 to 50 amino acids or nucleic acids have been added to your RNA, your sigma 70 will simply be removed. And your RNA polymerase will take over and continue to transcribe the messenger RNA. Now eventually you'll get other proteins, accessory proteins that will come in, and these will enable your RNA polymerase to dissociate from your DNA and will also lead to the release of your RNA molecule so it can go to the ribosomal RNA to make your proteins. And your RNA polymerase resets so that sigma 70 or some other sigma factor can guide it to another site. Now, termination of transcription in E. coli can only occur by two mechanisms. One is referred to as row independent, the P stands for row, and one is determined by row dependent. So for row independent, we are basically using palindrome sequences to form a hairpin. And so you're going to have complementary sequences form a hairpin, and when your polymerase gets to that hairpin, it's going to pause and it's going to fall off. Your row dependent is going to depend on a guanine adenine rich area that's called a rut. And then the rut is going to recruit a helicase, a row helicase, and this is going to migrate in the 5' to 3' direction. And this is going to have a dependence on ATP. So it has an ATP RNA DNA helicase activity. And this rut will stop the polymerase from further transcribing the DNA. Now for eukaryotes, this is going to be a little bit more complex. So we're going to have many different factors that control initiation. Some of our basal factors are going to be TATA boxes or cat boxes. Some of our constitutive, meaning they're always on, these are going to be housekeeping genes. They're going to have a GC box promoter element. Now the basal transcription factors are going to be controlling polymerase II activity, and these are always going to be transcription factor 2. TF stands for transcription factor. The 2 means it's RNA polymerase II. And so these transcription factors are going to help, similar to how sigma 70 helped the polymerase find its way to the region that needed to be transcribed. These transcription factors are going to help polymerase II find their way to a region that needs to be transcribed in eukaryotes. So here is a table of all of the transcription factors that are essential for polymerase II. And remember, polymerase II is responsible for making our messenger RNAs. It's responsible for making our proteins and enzymes necessary for our cells. And so we're going to have a TATA box binding protein, TBP, and that's going to bind to that TATA box. And then we're also going to have a series of transcription factors that are giving letters, and each of these transcription factors have a very important function. Most of it is going to be to get the polymerase to the area that needs to be transcribed so it doesn't transcribe an area that is not supposed to be transcribed. And it's also to activate that polymerase so that it can get going and making the protein that's necessary to be made. Some of them will have helicase activities. Transcription factor 2E and 2H will have helicase activities. And others are going to just simply be structure to help stabilize. Okay, so first, with respect to initiation transcription, we have our polymerase II, which is shown here, and it's always going to be associated with transcription factor 2F. Now we also have on our transcription factor 2F polymerase II complex is a C-terminal domain. And the activity of the polymerase occurs on the C-terminal domain. So we have our TATA binding protein, or TBP, which is going to bind first to this TATA box, and it's going to be associated with transcription factor A2, 2A. 2A is going to stabilize that TATA binding protein interaction, as well as the interaction with transcription factor 2B. 2B is going to bind to the TATA binding protein, and it's going to recruit the polymerase complex with transcription factor 2F and the C-terminal domain. And then your transcription factor 2F is always going to be associated again with that polymerase. Now all of this is going to be binding to that region that needs to be transcribed, and transcription factor 2H is what's going to have helicase activity to unwind the DNA. Transcription factor 2H is recruited by transcription factor 2E, which is also a helicase. Now, transcription factor 2H is also important because it's going to phosphorylate polymerase 2 within that C-terminal domain. So you're going to get phosphate groups added. And that's by that transcription factor 2H activity. And so here is this whole complex bound to that area that needs to be transcribed. And so you see here you have a sequence that is preventing transcription. So this is referred to as a pre-initiation complex. Now this pre-initiation complex will then open up and become a initiation complex or open. Now after about 20 or 30 base pairs, you're going to lose, I would say about 50 to 70 base pairs, you're going to lose transcription factor 2E and 2H. So their job is pretty much done. And then you're going to start to transcribe the gene. So your RNA is going to be transcribed. You have your phosphorylated C-terminal domain. You're starting to elongate your messenger RNA. You have elongation factors, which are simply proteins that are going to enhance the function of polymerase 2, and they have special names that we don't have to worry about. And then finally, you're going to have termination. Now, termination of transcription in eukaryotes is not very well characterized. However, there is a polyase signal, of course, at the end of each gene. There is a stop codon at the end of each gene, and so it's thought that they are going to play a role. So at the point of termination, you're basically going to release all your factors. You're going to release your messenger RNA, and then your polymerase complex with tissue factor 2F will recycle itself and go and look for other genes to transcribe. Now, one extra thing about transcription factor 2H. Now, remember, transcription factor 2H is very important. It's a helicase. It phosphorylates the C-terminal domain of polymerase 2. And it actually is going to enable the repair of DNA. Now, it does not do it itself. But remember, when your DNA unwinds and becomes single-stranded, that's when changes in the DNA can actually occur. So there can be a DNA lesion. And so transcription factor 2H will recruit the excision repair complex. And that's just another function of 2H. Okay, now finally, there are some things that can interfere with RNA polymerases. So we have some poisons or toxins that can do this. Actinomycin D is going to insert itself into the double helix, and it does this through GC bases. This will deform the DNA and not allow for that gene in that area to be transcribed. Acridine is also going to inhibit RNA synthesis in similar ways as actinomycin D. Ripampicin is going to inhibit bacteria RNA synthesis binding to the beta subunit of our RNA polymerase. Poisonous mushrooms are going to block polymerase 2 and polymerase 3 functions in our eukaryotes.